The Lampe lab at the Fred Hutchinson Cancer Research Center works on developing blood tests that can indicate if a person has cancer at a particular organ site in their body. Specifically, we are working to find so-called early detection biomarkers of colon, breast, pancreas, and lung cancer. Early detection of cancer can save lives, lower treatment costs, and by determining specific levels of certain proteins or antibodies allow for specific treatments or so-called personalized medicine. To discover novel biomarkers, we utilize glass slides that look like those one would normally use with a microscope. However, they are modified so they bind proteins called antibodies. Antibodies are the proteins in your blood that are produced by your immune system to bind to and help eliminate foreign particles and are what are produced in response to immunizations, such as the COVID-19 vaccine, to help fight off future infections. Antibodies are also manufactured to be used in many of the diagnostic tests performed in your doctor's office, since they can bind very specifically to their target proteins. Antibodies are typically drawn in a Y shape, and the top of the Y represents the region that binds the foreign proteins. We use antibodies to bind more than 3,000 different human proteins printed into little spots on these glass slides to determine the levels of these different proteins in blood. By labeling proteins with fluorescent tags, we can determine the levels of proteins, cancer-specific glycosylation events, or the presence of autoantibodies in the blood of study subjects and patients. Here, Jung uses a specialized robot to print the antibodies onto slides in a very precise manner. After placing 96 glass slides, he initiates the printing sequence. The printer head with 48 pins dips into the print plate and prints each slide 3 times before moving to the next slide. When finished, each glass slide can have 10,800 distinct antibody spots. By labeling the proteins in a blood sample with red fluorescent tags and incubating with a sample of reference proteins labeled with a green tag, Justin detects their relative concentration in blood. After loading this slide into a scanner, he shows us a magnified view of the fluorescence image of a slide. If a spot is red, that protein is higher in the cancer than reference. If it is yellow, it is unchanged, and if green, it is lower in cancer. We then have to extract the data from the array. Here, Dave manually curates some of the unusual spots identified by the software by deleting those that are problematic and reducing the area to be sampled in others. This image shows some actual data from a portion of the array. The panels marked B and D show protein levels in the array comparing a sample from a healthy subject and one with colon cancer. Panels A and C show the level of 2 cancer-specific glycosylation events. For example, the protein GCCR level in this cancer sample shows a 1.6-fold increase in protein, but a 19.3-fold increase in the CA19-9 over the healthy control sample. We can also detect the presence of autoantibodies. Here we show a magnified view comparing the level of AZGP1 protein, which shows little difference between case and control protein levels, but this cancer sample shows over 14-fold more autoantibody to AZGP1 compared to control. By comparing hundreds of people with cancer to hundreds of people without cancer, we can figure out whether a particular protein or autoantibody is a good candidate for further study in subsequent sets of samples. This process is called validation. Since people are quite diverse, many of the proteins that distinguish the first set of cancer and control samples will not perform well in the second set and will be discarded. Markers that survive through multiple sets of samples will then go on for formal validation by independent laboratories. The goal is to validate a panel of markers in blood for each of the major cancer types that would find cancer early enough to allow complete cures. We have made excellent progress with biomarkers of lung cancer. Lung cancer is the leading cause of cancer death, and heavy smokers are at increased risk for some subtypes of lung cancer. The major subdivisions are small cell and non-small cell lung cancer. Lung cancer early detection saves lives. Annual low-dose computed tomography screening has been shown to reduce non-small cell lung cancer mortality. Thus, an annual CT is recommended for heavy smokers. However, CT only detects the presence of nodules, and it is often difficult to tell whether they are benign or malignant. Since many smokers have benign nodules, we need to identify those that are malignant. Blood biomarkers could help with this. Also, CT does not readily detect small cell lung cancer or reduce its mortality. Blood biomarkers could also help with this. Our strategy for non-small cell lung cancer early detection is to combine plasma biomarkers with computed CT measurements and artificial intelligence-assisted CT image analysis to better differentiate malignant versus benign nodules. As an example, the purple arrow points to a nodule that is not easily classified. We are currently validating a decision rule based on the CT and biomarker values that assigns a probability as to whether a nodule is malignant or benign. Benign nodules would be followed in the next annual CT, while suspect nodules would be further imaged to better classify them. This project is a large collaborative effort with the University of Washington Radiology Department as part of the Fred Hutch Lung Specialized Program of Research Excellence Grant from the National Institutes of Health. Small cell lung cancer is a particularly nasty disease that is uniformly fatal unless it is detected early, but annual CT is not effective at reducing mortality. Small cell lung cancer causes production of autoantibodies that can be detected in plasma, and we have recently found a whole new class of these autoantibodies that can be highly predictive of the risk of small cell lung cancer. Thus, they can be used for early detection in a blood sample, or as targeting agents for localizing tumors via imaging, or to carry drugs to kill tumors. They could also be engineered to be targeting ligands for chimeric antigen receptor T-cell killing of tumors. Thus, we envisioned that a blood test taken at annual screening, if positive, could trigger immuno-PET imaging to confirm and localize the tumor for surgical removal and/or antibody-targeted or CAR-T treatment. This project is funded by a grant from NIH. Thanks for watching this presentation.